Hi, and welcome to the Biocompare Video Bench Tip. As always, our video bench tips are a chance for us to talk about developments in the tools and technologies available to life science researchers. Today, we're going to be focusing on the applications of optical imaging for in vivo safety screening studies. Optical imaging is a technology that measures light produced by optical reporters, which can be visualized through the tissues of a live animal using specialized imaging equipment and software. You can see examples of this when you are, for instance, using specific fluorescent agents and fluorescence imaging to quantify in vivo biological changes in disease models, or if you're using optical imaging to monitor various biological events such as tumor development and metastasis with cells engineered to express luciferase. While optical imaging is a mainstay technology for cell and molecular research, it is now finding its way into toxicology research. Toxicology research, which is of course critical for assessing the safety of newly developed drugs, generally relies on either generic physiology readouts or the measurement of indirect circulating biomarkers or even laborious ex vivo histopathology assessment. But new developments allow optical imaging technologies to be applied to toxicology. Fluorescence optical imaging offers the benefit of in vivo in situ imaging, allowing you to look directly at changes occurring in the target organs by using specific targeted fluorescent agents designed for in vivo use. Further, because fluorescent imaging is safe, it offers rapid quantitative results and corroborating ex vivo detection and quantification in tissues. With the right imaging agents, both organ toxicity and functional physiological tests can be performed in living animals. In this case, we're looking at kidney injury studies. Here, glomerular filtration rates, or GFR, is a measure of the ability of kidneys to filter waste products from the blood, and it can be significantly affected by drugs that have toxic effects on the kidney. Kinetic blood levels of a fluorescently labeled agent that measures GFR is cleared through the kidneys and can be measured in the blood circulating through the heart by fluorescence molecular tomography, or FMT. It is a simple procedure that requires imaging the upper chest of each mouse for 2 minutes to assess fluorescence changes in the heart over time. Drugs like cyclosporin A, known to cause kidney toxicity, alter the clearance of GFR fluorescent agents from the blood. This can be calculated as a 40% decrease in glomerular filtration rate in comparison to GFR rates in vehicle-treated control mice. Liver injury is measured by imaging live mice using an annexin-labeled fluorescent imaging agent that detects cell death. Acetaminophen, or APAP, is well characterized as a drug that can cause significant liver injury and cellular death. In this example, liver toxicity was measured in animals that received a single high dose of APAP and were assessed 24 hours later by injecting an annexin-labeled fluorescent imaging agent, followed by imaging the liver. 3D tomographic imaging clearly shows increased signal in the liver regions of APAP mice as compared to controls. 2D epifluorescence imaging offers a high throughput approach for in vivo imaging, and these highly significant 3 to 8-fold increases in signal were corroborated by direct fluorescent measurements in removed livers. The use of optical imaging for toxicology studies allows you to rapidly obtain quantitative data in a non-invasive manner with repeated measurements, reducing the number of animals and the amount of compound required for testing.